Hi, everyone. I'm excited to be joined here today with R&D engineer at LanzaTech, Sean Rollag. And today we're going to talk about his exciting PhD work on pyrolysis when he was at the Iowa State University under the direction of Professor Robert Brown. So Sean, how are you doing today?
You're pretty good, Andrew. Thank you. Thanks for having me.
Yeah, thank you for coming. I love this. I love talking to other scientists, and what you're doing is near and dear to my heart in pyrolysis. I actually started, that's what got me interested in chemical engineering. So super excited today to talk about, who you are, how you got into this, and what this work specifically kind of led to, and where you see the future going as well. So thanks again for coming on the show today. So today we're gonna talk specifically, and I'll have a link to this in the video, but today we're going to talk about your paper, which was titled "The Role of Catalytic Iron in Enhancing Volumetric Sugar Productivity During Autothermal Pyrolysis of Woody Biomass." So I guess first, before I jump into kind of how you got into this, what does that mean?
Oh, well, the goal with the research was just really driven by the idea of process intensification, of trying to get as much productivity out of just a given reactor or given chemistry as you can. Just trying to build as small as possible, which allows modular manufacturing. So if we could produce as much as possible in a given volume, that was the benefit we were really going for, so we can containerize our reactors.
I see.
Number up over scale up for that.
I see. So I see this term intensification thrown around a lot. So that's making things happen faster so you can go smaller. Is that right?
Yeah. And a lot of the funding came from the Rapid Institute, which is all about process intensification and research. So that was the main driver. And this was an avenue we found where you could intensify the pyrolysis process quite significantly for certain feedstocks and certain products.
Okay, great. Well, I'm excited to jump into that, but kind of before we do, how did you get into this field?
Well, my kind of initial introduction was actually I grew up on a farm, and so there's so many challenges that have to be solved there just day to day that kind of from childhood as I was tearing stuff apart and putting it back together, just trying to understand how it functions at the basic level. And once you know that, you can kind of build off that and do all kinds of crazy things. That introduction to more chemical engineering was actually I read a book called The Alchemy of Air, about the production of the Haber-Bosch process. I thought that was just so fascinating how they take this really simple kind of chemists developed thing that made a couple of drops, and how that had been scaled up through engineering and kind of catalyst research and stuff like that to generate, basically build the modern world of agriculture that we have and how we feed everyone. That kind of inspired me of, I want to take an idea and actually make it real and change the world sort of thing.
Yeah, and of course, Haber-Bosch did do that, right? They fed the world. So. Yeah, very cool. Well, so this work in particular, so this is pyrolysis work, intensification. What was, you talked a little bit about this, but what was the motivation behind, picking this direction and exactly what you did?
Yeah, so my personal motivation was kind of growing up on the farm, and how do you help kind of the rural economy add value to what is really trash, the corn stover, stuff like that, the egg waste, or just environmental as well of, the rains aren't coming at the right times. That really impacts farmers, and you can directly see that the weathers are changing. You can't really plan out the seasons and stuff. We're watching, the corn belt creep further north as it gets warmer and warmer up there. So firsthand experience of climate change, and then how do we actually work on that is through renewables or stuff like that. So that's really, I wanted to be involved in that industry. And I think pyrolysis is a really good avenue for really good climate solutions to produce the fuels and chemicals that we day-to-day lives.
Very cool. So as a farmer, you got to see some of the effects of climate change, even this far north. In
firsthand, the main thing was that the growing season is getting longer because of the freeze.
Yeah, so that's really cool. So as a farmer, you saw this climate change effect, and it kind of drove you to get excited about doing research to help.
Yeah, I
guess mitigate or help in some way.
Again, it was a problem. I just want to find a solution to problems. So I'm a problem solver. That's what engineering is, in my opinion. It's just solving different problems.
I can relate to that for sure. So what's really unique about what you guys were studying here versus what everyone else has done with? Because of course pyrolysis has been done for a while, and
Mhm. So I think the big development was actually a previous grad student to me, Joe Poland, had developed this process of autothermal pyrolysis, where you're adding, rather than conventional pyrolysis being just completely inert conditions, if you slip some air into there, you can combust some of the products to generate the energy you need to drive the pyrolysis reactions. And that really allowed that, again, that process intensification of if you're generating heat internal to the reactor, you no longer have this wall they have to try and transfer heat to, so you can scale the thing up. And
yeah,
you don't have all this heat you got to try and get to the center of the reactor, you can generate it there. And so he just kept feeding more and more biomass in because you can dump more and more air in, but then the temperature goes up. So you have more and more biomass to cool it back down. As long as you keep them balanced, you can really increase that throughput of that reactor. So when they had done that, then it really unlocked, we can make a ton of bio stuff, but what could we do with that? It's got a bunch of phenolics in it. There's this trace amount of sugars that maybe we could do something with that if we get more sugars in it. So then the research turned into how do we make more sugars from pyrolysis. And so that was kind of when I was brought in, which they started to encounter some issues with the more traditional treatments out there. Nitric acid.
so trying to increase the amount of sugar yield, and that's important because that's a feedstock to some industry, or
the cellulosic sugars have a lot of high value because you can ferment them into a bunch of different products. There's all this research out there on taking sugar and fermentation into all these various products and commodity chemicals and stuff. Like ethanol is kind of the main one now, but you can bioengineer those bugs to grow very different compounds from those sugars, but you need that kind of building block of sugar molecules in the first place. And the enzymatic hydrolysis approach has some of its own issues. So we're trying, maybe it was another way to get sugars out of biomass. It's there, but it's not much. But pre-treat it, you greatly increase that. So it kind of started to look attractive. So it was an area that was, I think, ripe for some development work to be done.
And it looks like what you did was quite successful. You increased yields from 5 to 20% in some cases almost of those sugars. That's pretty cool. What were the biggest challenges that you came across while you were doing this research?
Well, there's a number of them. The biggest one was actually kind of the scale up, because it's easy to do the micropyrolysis experiments. You can do dozens of those a day if you want, but when we started to scale it up, the actual retreatment wasn't working initially, and it was a whole bunch of just designing stuff that was more of a kind of more shakedown testing was required on, how long does it take the chemicals to actually diffuse into these biomass surfaces or something, because we were dealing with the powder that's not so considered. So we struggled with that for quite a while until we figured out kind of the practice of how to do this at scale, and then a lot of the effects of the reactor agglomeration that we're really trying to avoid. You can't see that until you get to an actual continuous reactor system. So there's a lot of struggles of how do we scale this up. We need enough expert, also small enough where we can do it multiple times a day.
Yeah. So you actually did scale this up. So you went from this micro powder scale. How big did you end up going?
The largest scale that we got to is actually a pilot scale. We did half ton a day. Every a whole day. That was hundreds of kilograms of biomass. So that was a lot of fun to see this idea, and then all of a sudden you can hold bottles full of the oil coming out.
Yeah, and what a cool experience to be able to do that in grad school, because my experience never got bigger than that. A few micrograms. So that's pretty
unique
neat.
Approach of the Brown research group was that ability to scale up to pilot scale testing. So
yeah,
definitely has advantages.
And I've obviously worked in pyrolysis a bit. I'm curious to learn, and I've seen it just talked about more and more these days as decarbonization is brought into the limelight. I'm curious to understand what you think pyrolysis, where its role might be in climate change and decarbonization, and just your thoughts around that.
Yeah. I guess before I had gone to grad school, I never really heard much about pyrolysis. It's not a big field that people talk about very much, but I think it's gonna get its day in the spotlight. For mostly is you can produce a lot of you want to replace the whole barrel of oil and everything, so you need to make more than just ethanol. You can make all these other chemicals. So through that fermentation of sugars, you can make all those different chemicals for producing, different clothing and stuff like that. So I think the advantage of pyrolysis is you can make those sugars and do it in a carbon negative way. So as we get further and further into, global climate change and stuff, where there's too much CO2 already in the atmosphere, we need to start pulling some of that out of the atmosphere. And I think pyrolysis allows you to do that by giving you this, mostly it's the biochar stream. If you can sequester that, now you have a net negative flow of carbon out of the atmosphere. I think some of the other hot topics right now, like the electrofuels and stuff, where you're pulling CO2 out, using electricity to try and turn that into a fuel. At best, that's neutral. But through pyrolysis, you can get negative. You can start drawing down that carbon in the atmosphere to meaningful levels, where kind of reverse some of these effects we're seeing with climate change.
Yeah, I love that. And obviously, I'm on the analytical side, so I want to shift gears a little bit to the analytical challenges that you had. Specifically, what did you have to measure in these experiments? How did you do that? What were the challenges in doing that specifically with pyrolysis?
So, most of my research, the fun part was obviously the scale up, but you have to start small. So there's a lot of micropyrolyzer into a GC FID system. Had the polyarc system on there to really help us with our quantification of all these different compounds, because pyrolysis produces just a soup of different molecules. And so you can't go through and try and quantify and build calibration curves for every single one of those. So the way we would use our system is have a mass spec and then the polyarc FID system, so that we could identify what it was, and then use polyarc normalization to methane to actually quantify to some accuracy what had made of all these hundreds of different compounds. So it really allowed us to save a ton of time building calibration curves and stuff. There's all the unknowns. We've got a different catalyst, and all these unknowns have changed in different ways. So what ones are effective, what ones weren't affected by it. So that rapid quantification of different products is very useful. And then with our kind of lighter fractions, you end up with very oxygenated molecules, which you can't really detect very well with that FID. So again, having that polyarc system allowed us to look at kind of the lighter formaldehyde, stuff like that, the really light organics that with certain catalysts, you make a whole lot of them, but if you can't see them, it's tough to actually quantify how much. So definitely had that. The difficulty we had with that system is the levoglucosan molecule, which is our main target, is such a high boiling point molecule that it's very difficult to get that out of the column after every test. So you run for like a week or so, and then you'd have to basically clean out your liner and everything, and you'd filled it up with some really heavy tars and stuff. You have to flush your columns all the time. It's kind of tough. That was the molecule we cared about the most. It was the toughest one to really get a solid baseline on, because it just wouldn't go away after from.
Yeah, you're kind of right on the limit of GC, getting into the liquid chromatography regime. Yeah. I get that. How hot were you going in the gas chromatograph? Did you get up to the 400 C?
Column. I think we got up to like 200 plus C on some of them.
Yeah.
I think it was like 300 C. So
yeah,
harsh conditions. I mean,
yeah, you start getting.
Wouldn't be too happy with GC guys if they're what we were doing sometimes too.
Yeah. Yeah, those GC components start to not get too happy when you get really, really, really hot. You start to bake off that polyamide, and on the column it turns brown. And I hear that. Yeah, were there any other kind of big challenges that analytically, in terms of getting the data you wanted, and maybe a follow on to what you said with the polyarc, were you able to get near complete or complete carbon closure when you use the polyarc? Do you have a good idea of where your carbon's going?
We would get fairly high carbon closure. The issue with the pyrolysis is you'd still end up with dimers and trimers that just, you're not going to get that. You're like,
yeah, it's in your inlet liner.
You're not getting those either. So we had some issues with some of the carbon closures. Some of the pretreatments were a lot better than others, though, of actually fractioning everything into monomers quite well. Some of them that left everything as trimers.
Yeah.
And stuff. Those are kind of possible. The liquid chromatography couldn't handle some of those compounds.
Yeah.
That quite a bit, but yeah, we had some pretty good results. The lighter compounds, and once we kind of got the system down, it's just the methods we need, stuff like that. One of the things that we never quite figured out is there's always a mismatch for some reason between how much levo we quantify and how much glucose we could hydrolyze from our samples. We always end up with more glucose than we put levo in. So there's always this unknown of, is there a dimer or something that's just not coming out of the GC that's getting hydrolyzed to glucose. That's why we, in the paper at least, we try to report everything on kind of the glucose basis. The practical sugars, but if you were to commercialize it, that's what we'd end up with, is this much glucose. We couldn't quite quantify that on the small scale because there's a mismatch there. If it's coming from somewhere, but we never quite identified where.
Yeah, so it sounds like there's still some challenges yet. We need someone to invent the GCLC that can do everything.
Yes.
Dimers, trimers. Yeah, I know. I thought about it, but we're not there yet. Well, I love that, and it sounds like the polyarc was helpful, which I love to hear that. That was, that makes me happy, of course, that we have customers out there using it.
We had 2 polyarcs in our lab, and there was some fighting amongst grad students, so we got to use those GCs because those are the coveted ones. You had to have a good argument of why I need to use that one because I need to identify all these compounds. Or if you're looking at just at one compound, it's like, no, we got a calibration curve for that one. Don't get it. You spend time on that instrument. It's not important enough for that. So definitely needed more of those in our lab.
Yeah, it sounds like we got to sell you guys more. Well, I'll go back to Brown and talk to him. So you had a lot of great success with this, and obviously the yield gains were huge. What questions do you think still remain, and kind of looking into the future, what do you want to see next?
There's definitely still some questions remain, especially at more fundamental side of things. We identified this trend of kind of the S and G lignin being very important for determining why there's a separation of hardwoods, softwoods, which has melt and stuff like that. More fundamental research into what is the mechanism behind why these 2 lignin molecules or monomers behave so vastly differently with these catalyst ratings and stuff like that. Some of those it's a correlation we identified, but the reasoning behind it is definitely a fairly large unknown. We did look into it, and there is another paper published on that, but I don't think we would find it. I think there's still a lot of answers out there for that question. What true fundamental effect are we seeing? Just kind of got a correlation.
And is that complicated because there's just so much going on in these reactions? It's not A to B reaction.
We asked some of the DFT model guys, can you build this dimer? And that's really computationally complex, and trying to figure out how the very different catalysts were interacting or what state those catalysts were in. It's quite a challenge, especially with the complexity of the biomass system. Once you add the cross-linking of the hemicellulose and cellulose fractions, and obviously not steady state isothermal conditions, you got all this changing fluctuations and demilitarization. It's a very complex system to try and model, but I think you need to be able to probe it at that level to actually understand what's going on.
Yeah. And this question is more for the students out there who are making that tough decision as to whether they do a PhD or go into industry right away. What would you tell other students who are looking to get into this field or potentially do research here?
I would say you really got to want and a love for science, because it consumes a lot of your life. It's not, it's not 40 hours a week. It's, it's 7 days, 24 hours, because you'll be, going to sleep and you'll have an idea. You gotta, write it down quick, because it's constantly, you can never really turn it off, because it's all consuming at times. There's some really slow periods where, you have 30 tests and then not just having any success at all. And then you have one test and then it just branches off into all these other discoveries and stuff. So it's, you gotta love the slow times as well. And, I'm still learning things. It's not exciting stuff, but every day I'm learning something that was unknown to the world before. So that kind of mentality to drive you, or it's better to just go into industry right away and have some fun there rather than the research route. It's definitely, it's a process. You gotta want it though.
Yeah, I agree with that. I think PhDs we're wired a little bit differently in terms of maybe our tolerance for pain. I don't know, but
I had a number of people that have asked me years now, oh, I'm kind of thinking about it. I can't find a job yet, so maybe I go to grad school, buy myself some extra time to find a job. That's not the motivation. You want going. You'll be in there 6 months and maybe you'll find a job, and then it's like, oh, I don't want to be here. Why am I wasting my time in grad school?
Yeah, well, I think that's great advice. This will be valuable to students and PhDs and people in the field, as well as hopefully some people who are not scientists as well. So appreciate your time, Sean. This is fantastic, and keep up the good work. It sounds like you're in industry now and changing the world in other ways. So
yeah,
we'll keep watching what you're doing.
Hopefully I do get to make some big changes to the world. Is still working on it. So
Awesome.
Thanks for your time as well. I enjoyed talking about my research, and I'm glad people want to hear about it. So.
All right. Thank you so much to our guest, Sean, for your time today. It's been wonderful. You did a great job, and I think doing some really interesting stuff there. And we'll keep an eye out for all the great things that you're going to continue to do. So thank you, Sean.
Thank you.